So in terms of honeybees, there's been a few different ways that environmental DNA has been used to study them. So through honey, through the pollen from the adults themselves, from debris that's left in the hives, and more recently, by use of air. For bees that are more solitary, obviously they tend to gather in a lot lower abundance, but also bees seem to not shed DNA quite in the same way that other creatures do. However, there have been studies using eDNA, whether this is through soil or, again, more recently, there's been a big study about collecting petals and trying to find contact DNA of insects that have landed on those. Of course, without visual study as well, you don't know if bees have landed on those flowers. So the bees are targeting the flowers and their communities rather than targeting the bees themselves. In order to target more solitary bees, usually this is done by either hand catching or pan traps, or recently bee homes have been used in order to gather the larvae from them and use metabarcoding in order to look at the DNA that's present there. And obviously, these are usually lethal methods of looking at ecology of these bees. So what I'd like to do is I want to see if the biological material that is already present in the bee nests and see if we can collect the same information from the biological material that is there rather than using the larvae themselves, basically to try and non-invasively look at the ecology within the bee home. So how I did this, I placed 100 bee homes around Guelph and Cambridge. This was over the summer flight season in 2022. So all the homes were collected in September, hoping that they would be occupied and they'd be filled. So I took all these back to the lab, I cut open the bee homes, and then I took out the larva and then its associated cell contents. And I did this for every single bee brood cell that was present within those samples. In total, I ended up with 1,483 paired samples of a larva and its associated cell contents. This was then sent to the Center for Biodiversity Genomics at the University of Guelph, where they did DNA extraction, PCR amplification, and sequencing for 4 different groups. So for the plants that's present, fungi, microbes, and of course the insects that are present within the bee homes. So once I got that data back, I ran it through the KIME2 and Data2 pipelines. And if you're interested in this, I tried to use more created databases, but these were the databases that I used in order to identify the ASVs. So which larva is present? So in order to determine this, we defined occupant as the dominant read count above 1,000. So what ASVs present had above 1,000 reads. So 2,190 of my samples had occupants identified. This is roughly 74% of the samples that I sent away. Of those, 1,158 occupants were identified as different bee species. The other samples tended to be other different insects, so lots of wasps, moths, earwigs, that kind of thing. So of these 1,158 occupants, 990 were in a larva cell pair. So I had 495 pairs of samples, and these spanned across 95 different brood tubes. So how similar are the larva and cell content data? So there's 2 graphs here. So each column shows the proportion of shared and unique detections between brood tubes. So black indicates the proportion of ones that were genera that were detected within both cell content and larva samples. Green is unique to cell and blue is unique to larva. So the 4 columns on the left, they are overall detections, everything that we detected within the samples, whereas the right columns are all ones where we've excluded the rare taxa. So taxa that were only detected one time, like genera, that were only detected one time in the entire data set. So I included both because there is actually a difference once you exclude the rare genera. It's most noticeable in the fungi, which is the leftmost columns of each graph. And if you look at that, you can see that the proportion of shared detections jumps from 50% to almost to around 75%. So we're seeing a higher degree of these of taxa in both cell and lava samples. So to kind of build on that, so here's a bar graph showing the top 10 plants detected in lava and cells. And as you can see, they're extremely similar. So not only are we picking up like the same genera, the same top 10, they're all the same. And then they're also roughly the same proportions. Like, yes, we do have more detections within the larval samples, but the proportions of the samples themselves, we're getting the same thing. So it makes for a very satisfying, colorful graph. Anyway, so this is great because it means that we can pick up like the main plants that are present in the bee homes from using the cell content and the lava. So I did do obviously the plants, insects, fungi, and microbes, but I'm just going to focus on plants for time. But we are seeing a similar pattern across all of those different groups. Looking at the community level. So this is a principal coordinate analysis. So it's just a way of visualizing the data in the 2D space. So green is the cell samples and blue is the lava. And as you can see from the holes, like you're seeing like a high overlap between the community that we're recovering from the cell samples as we are from the lava. When we considered this statistically, we found that sample type explains very little of the variation that we're seeing, less than 1%. So it means that in both sample types, we're recovering most of the community that is present. So if you are looking into likely plants that have been provisioned by the parent, either for food or for to build the cell, then the cell content can give you the same insight as using the larvae themselves. So hopefully it could be something that's non-invasive and we could use further down the line. Okay, so in summary, we found that 85% of the time we detected the same occupant in cell and larval samples. This seems to be a way that we can reliably recover the community of plants in both sample types. And as I said before, the same thing we're seeing for fungal and microbial communities. So this is an eDNA approach that can work on native pollinators potentially on a large scale with high efficiency. So the biological material in bee homes can be used as a non-invasive tool for quantifying the ecology of cavity nesting bees. It's something we can use that would be non-invasive, which would be great because you don't want to harm the bees. And for future studies, the hope is that we can use cell contents after emergence, so the leftover material that's within the bee home brood tube, to quantify the community ecology of the larva non-invasively. And yeah. So quick thank you to everyone who's helped my research. There's been plenty of people involved, but especially everyone in the Claire lab, because they're always quick to lend a hand and offer great support. Okay. Thank you for listening.